Hey everyone, in this video we're going to be going through the whole of Edexcel GCSE Biology, Cells and Control. So we're going to be looking at things like the nervous system and the brain and loads of other stuff. If you'd like to follow along with this video, over on my website you can download my notes and my free revision checklists. So mitosis is the process of cell division. And it's really important for growth and repair. So before mitosis even starts, interphase needs to happen. And cells spend about 90% of their time in interphase. During interphase, the cell needs to replicate its DNA. The cell also needs to make lots more of its organelles like mitochondria and ribosomes ready for the new cell. Mitosis actually begins with prophase all the way up until telophase. During prophase, the nucleus breaks down and spindle fibres appear. During metaphase, the spindle fibres pull the chromosomes to the middle of the cell. And you can think M for middle. In anaphase, these spindle fibres pull the chromatids away to each end of the cell. The chromatids are just one half of a chromosome. And you can think A for away, and then during telophase the nucleus reforms. The last stage is cytokinesis, which is where the cytoplasm finally splits, creating two daughter cells. Now it's important to remember that these cells are always identical. So mitosis always produces identical cells. So mitosis means that the DNA is copied millions of times in the body, and if something is copied millions of times, there's bound to be a mistake. These mistakes are called mutations. Mutations in the DNA can cause mitosis to happen uncontrollably, creating a tumour. So growth means to increase in size or number. Plant cells usually elongate and divide at the roots. Elongation just means the cells stretch right out to help it get bigger. Animal cells typically just divide. We can use percentile charts to measure growth. If the organism is in the 100th percentile, they are well above average. If they're in the 50th percentile, they are average. And if they're in the 1st percentile, they are well below average. It's a way of comparing the organism's growth against other organisms. So if they were in the 50th percentile, this means 50% of the organisms are smaller than it, and 50% of the organisms are bigger than it. Stem cells are unspecialized cells that can differentiate into specialized cells. So an unspecialized cell is one with no particular function. It has no job, it's just a cell. Differentiation is the process where these unspecialized cells turn into specialized cells. They switch particular genes on or off so they can develop the desired characteristics. And then a specialized cell is one that is adapted to do a particular job. For example, a nerve cell is very, very long to help carry impulses over long distances. There are three types of stem cells. Embryonic stem cells are the ones found in the early embryo. These can turn into any type of specialised cell in a human. This is because these are the original cells we start with, so they need to be able to turn into all the cells we need. The second type of stem cell are animal stem cells. And these are already partially specialised, so they can only turn into certain types of cell. For example, a stem cell found in the bone marrow can only turn into the different types of blood cell. It wouldn't be able to form a brain cell or a muscle cell. This is because animals stop growing, so they don't need to be able to create new cells all of the time, only the ones that could need replacing. Now plants have meristem cells, and these can turn into any type of cells in a plant. This is because plants continue to grow throughout their life, so they always need new specialised cells. We can use stem cells in medicine. If we stimulate them to produce the cells we need, we can help cure and fix certain diseases. However, there are a few problems. The cells could continue to divide and create a tumour, or the body's immune system could reject the cells, killing it off. So the nervous system is the way that the body sends and receives information. There are two parts. The central nervous system is made up of the brain and spinal cord. And then the peripheral nervous system is made up of all the other neurons. There are three different types of nerve cell. The first is sensory neurons. We have a dendron, an axon, as well as dendrites and axon terminals. This middle part of the cell is the cell body, which is where the nucleus and all the other organelles are found. Sensory neurons are connected to our receptors. These are the things that take in the surroundings, so our senses. All sensory neurons are in the peripheral nervous system. The next type of neuron are motor neurons. We still have our axon, however we have no dendron. Instead, the dendrites are directly on the cell body. And then we still have our axon terminals. Motor neurons carry impulses to effectors, so these are our muscles or our glands. The final type are relay neurons. These are short neurons found in the brain and spinal cord. These look exactly the same as motor neurons. We still have our axon, dendrites on the cell body, and our axon terminals. So you can see these covers over these neurons. This is called a myelin sheath. These are there to insulate the neuron and speed up the impulses. So an impulse always begins with a stimulus. Our receptors then pick this stimulus up. The impulse travels through all the different types of neurons and through the brain before connecting to our effector, which is our muscle or gland. This in turn then signifies a response. So in this case, the stimulus is a dangerous animal, so the snake. Our eye sees this and picks it up and sends an impulse for our sensory, relay and motor neurons. It then connects to our muscles, which then contracts to help us run away. So between neurons there are small gaps called synapses. These are about 20 nanometers wide. In this gap, neurotransmitters are released. Neurotransmitters are only released from axon terminals, so this means the impulse carries on going in one direction. The next neuron then picks it up with its dendrites. This means that a new impulse is generated at every neuron, so the signal doesn't lose strength. So there are a couple of parts of the brain you need to know. The cerebrum makes up the main bulk of the brain. It's divided into two hemispheres, which are exactly the same on each side. In the cerebrum we have 4 lobes. At the back of the brain we have the cerebellum. This is responsible for our motor functions like walking. And then at the bottom of the brain we have the brainstem or the medulla oblongata, which is responsible for our unconscious processes such as breathing or controlling our heart rate. So the brain is trapped inside of our skull, so it's really hard to access it to either look at it or treat it for problems. We need to use scans to be able to get an idea of what's going on inside the brain. The first type of scan is a CT scan. This builds up an image of the shapes and structures in the brain. An X-ray beam passes around the head in a circle, and then detectors measure how much is absorbed by the brain. This then produces an image of the brain that we can look at on a computer. The second type of scan are PET scans. This measures brain activity. The patient is injected with radioactive glucose. The brain cells take this in for respiration. More active cells are going to take in more of this glucose, so then we can use a scanner to measure how much each part of the brain takes in. So in the brain we have a reflex arc. And this is designed to help the body quickly react to potentially dangerous situations. It's a completely unconscious process. It means that any dangerous signals can loop straight round and trigger a response, instead of having to stop and waste time going through the brain. Having no brain involvement means that this is much quicker. So this helps with our reflexes. So the eye is our receptor to help us see. There are lots of different parts of the eye to help us do this. At the front of the eye we have the lens, and this lets light into the eye and focuses it. To control the lens and how much it bends light we have ciliary muscles. We have the iris, which is the coloured part of the eye. The hole in the middle is the pupil, which allows light in. We have the cornea to help focus light. We have the optic nerve to send this vision information to the brain. And then at the back of the eye we have the retina. The retina is the light sensitive part. It's filled with two types of cells, rods and cones. Rods are responsible for detecting differences in light intensity. So this detects our shades, so light and dark. This means rods work really well in dim light. Cones are responsible for detecting colour. These need really bright light to work. So focusing an image is mainly done by the cornea. This refracts the light rays to bring them together at the back of the eye. So I said that the ciliary muscles help make the lens fatter or thinner to focus this light. If an object is far away, the lens will be a lot thinner to focus the object, compared to an object is close, which would need a much fatter lens. So if the person has short-sightedness, distant images appear blurred. Light is forced in front of the retina, so we can't detect all of the image. This is caused by the eye being too long or the cornea being too curved. If the person has long-sightedness, then close images appear blurred. Light is focused behind the retina, and this is caused by the eyeball being too short or the cornea being too flat. So cataracts is a problem that makes the vision cloudy, and is caused by a protein build-up in the lens. To correct this, an operation can be done to replace this lens with a plastic one. Colour blindness is another problem with the eye. It's when the person can't see particular colours and is caused by faulty cones. Red-green colour blindness is the most common. We can use tests like this to help see if the person can differentiate between two different colours. If this video helped with your biology revision, please subscribe to my channel and check out some of the other videos I have.